Would it surprise you if I told you that the cells of your nervous system that transmit signals, that are responsible for your coordination, that are responsible for your memories, that are responsible for your eyesight, your taste, smell, only accounted for about 20% of your whole nervous system? Would that surprise you? You would think that that's the business end of the nervous system, but in fact 80% of your nervous system are supportive cells that are referred to collectively as your neuroglial cells. Neuroglia literally means nerve glue. And there's a bunch of these. They're vacuum cleaners in your nervous system that are called microglia. There are cells that help create the blood-brain barrier and pass nutrients and waste back and forth from the blood system to the nerves. They're called astrocytes. But the neuroglial cells I want to talk about right now are insulating cells. And these insulating cells are called Schwann cells or oligodendrocytes. Now, this is another good compare-contrast type question, because both Schwann cells and oligodendrocytes will myelinate axons. Have you ever done any electrical work or seen anyone doing work with switches or ceiling fans, and you've got these wires and you've got to twist them together, and then you get that black electrical tape? Have you ever seen a real good electrician? He'll wrap the tape around maybe one, and then he'll stick his finger in that hole and then just go bzzz, just put several layers around it. My dad, when he does electrical work, he has big knots of that stuff because I guess he doesn't want the electricity to get out. I don't know why. My dad always says if a little does a little good, a lot will do a lot of good, which is not necessarily the case every time. But in this case, these cells do the same thing, except they're not using plastic tape. They're using their plasma membranes. And so layer upon layer of phospholipids, with all the cytoplasmic material being squeezed out, all these layers after layers of phospholipids make up what we refer to as myelin or the myelin sheath. Schwann cells are going to myelinate neurons in the peripheral nervous system. Whereas oligodendrocytes that look kind of like an octopus, they myelinate neurons in the central nervous system. You'll never find them going back and forth. It's always about where they're located. Schwann cells will each myelinate a segment of one axon. But since oligodendrocytes have multiple processes, these can myelinate multiple segments on multiple neurons. So do you see how they're similar and yet they're different? So here's an oligodendrocyte, and you can see this process has wrapped this axon. This process has wrapped this one. There's another one and another and yet another. So multiple segments of myelination wrapping around and around and around and around the axon. Notice I'm saying axons. You will not myelinate the cell body, and you will not myelinate dendrites. Just the axons. So myelin sheaths, this is simply that covering of myelin all along the length of our axon with little gaps regularly spaced in between the areas where we have myelin. These are called the nodes of Ranvier. So here we have a Schwann cell starting the process of myelination. And here that cell is completely wrapped around maybe 30 or 40 times. Its plasma membrane, all the cytoplasm gets squeezed out to the edge. And then we'll have a space. And then we'll have another Schwann cell that does the same thing. Space, another Schwann cell. So that whole length, that is our myelin sheath. Here we can see our myelination interspersed with our nodes of Ranvier all along the length. This particular axon, we've got Schwann cells that are associated with these axons and a number of different ones. But notice they aren't wrapping their membrane around. They're just kind of hanging out beside them. This is an unmyelinated axon. And the myelin is just simply those 30, 40 layers of phospholipid, not just the Schwann cell being associated. All right? This is a transmission electron micrographic view of an axon cut in cross-section. And you can see this dark ring that goes around. You can see how dense this material is, and it's just layer upon layer upon layer of the plasma membranes of those Schwann cells. That's the insulation, the electrical tape that's wrapped around. This is a watertight barrier. It's a molecular barrier. Nothing's going to get through that. Now, here's the cool thing about these myelin sheaths. We see them dispersed out, and we see the nodes of Ranvier. This is the part of the axon that's exposed, that's not myelinated. And we see that nodes of Ranvier occur at about 1 millimeter apart. Now, does that distance, does that mean anything to you from what we talked about earlier today?
Like you have sodium ion channels within that.
Sodium voltage gated channel. Sodium comes in. Where does it go? Everywhere. And how far can it travel? 1 to 2 millimeters. So if we have our nodes of Ranvier spaced 1 millimeter apart, then guess what? This sodium voltage gated channel, it will open and sodium will travel that 1 millimeter. Maybe there have been hundreds of other sodium voltage gated channels on an unmyelinated axon that had to be activated, but did they really? Because that sodium is going to be enough to influence, let's say you're the next node of Ranvier. That sodium is going to flood under here so rapidly, it's going to depolarize you. There's no need for the stuff in between. So the nodes of Ranvier and the myelination are going to conserve energy in the sense the cell doesn't have to make as many voltage-gated channels. But guess what else? Without those voltage-gated channels, ions aren't going to be transported here. And so you're not going to have to use as many sodium-potassium pumps. And those are the suckers that use so much energy, right? So you have the voltage-gated channels and the sodium-potassium pumps concentrated at the nodes of Ranvier. So you save energy. But not only that, by stimulating an action potential at the very beginning of the axon, we refer to this as the axon hillock. That's going to be important. This is where we have the highest concentration of sodium voltage-gated channels. You don't have voltage-gated channels anywhere else in the cell, not the body, not the dendrites, but you have a ton of them at the axon hillock. And when you stimulate that action potential, here, sodium comes in, it's going to flood all the way over and open the channels here. Sodium floods in here. It'll flow with cable properties, causing a depolarization here. So, do you see what's happening? It's as if our action potential is not just flowing like the wave like we saw at our football game, but now it's as if the signal is jumping from node to node to node. It used to be called saltatory conduction. But it doesn't, in fact, jump. It's just the ions travel with those cable properties so rapidly that by myelinating an axon, concentrating the receptors, those voltage-gated channels, and the sodium-potassium pumps at the nodes, you can increase the transmission of that action potential from 1 meter per second to over 100 meters per second. That's fast. The fastest human right now is Usain Bolt, I think. The 100 meters in just under 10 seconds. Yeah, this is 10 times faster than Usain Bolt. This thing can do 100 meters in 1 second. Pew. That's impressive. I don't care who you are. That's impressive speed. And this is happening in your body every millisecond, your entire life. How fast these signals are being transmitted. So again, with our myelin sheaths, we see we're having our depolarization, repolarization, hyperpolarization resetting only occurring at the nodes of Ranvier, speeds up the signal, saves us energy. We don't have to make as much stuff. Man, that sounds like a great plan to me. That is really cool. So I want to present this question to you. Talked about my dad making that big, huge knot of electrical tape. In the case of neurons, the larger the diameter, and I've read in some articles, and I don't really understand the physics behind it, but the larger diameter of the axon, the faster the transmission of the impulse. Thus, the myelination, the thicker the myelination, simulates a larger diameter to the axon, so the thicker the myelination, the faster the impulse. I don't quite get my brain around that. But when we look at axons that are important for your survival, and we find that those axons have the thickest myelination, it sort of intuitively makes sense. So when you think about your body, and you think about the nerves that are going to be the most critical for your survival, which do you think is the thickest nerve, is the most heavily myelinated nerve? Vision. Vision is your key sense to survival. It's going to help you find food, it's going to help you find a mate, and it's going to help you not be somebody's breakfast, right? Now, hearing is important. Smell is important. But vision, that's the key. Now, I'm a country boy. We got some other country folks. You're city. You're Lufkin, right? That's city girl. Okay, I'm a country guy. And I have walked along many a dirt road in the evening or at night, getting home from when I was playing as a kid. And if you're walking, or maybe down an alley or something and it's dark, when you hear something that sounds scary, you're not just walking along and you just go, oh, there's something big and hairy right over here. I don't know what it is. I'm just going to keep. No, that's not what you do, right? No. What you do is when you hear something big and scary, you immediately, you're focusing your vision on that thing. Because you want to know what it is. You want to assess, what do I have to do? The fight or flight thing, right? We're going to get to that autonomic nervous system next chapter. I don't necessarily do that. Most of the time, I'm doing like this, looking back as I'm running. So vision, your optic nerve, it's the thickest nerve in your body because it's so critical to your survival. What about pain receptors, these sensory neurons? Do you think they're myelinated? You betcha. Heavily. Again, with our autonomic nervous system, it sounds like automatic. You ever touched a hot stove? How long does it take you to move your hand off that hot stove? You move it and then you realize what happened. That's how fast. Because it only takes just a very few seconds to get third-degree burns. So, I mean, that's a built-in reflex. It's super, super critical. What about motor neurons that myelinate your involuntary muscles? Your smooth muscles of your digestive tract. Are they myelinated? What do you think? Yeah, but not so much. Not so much. You're never going to have a major emergency because you didn't digest that Big Mac fast enough. Okay? I keep saying Big Mac. I need to say orange chicken. I had some orange chicken for the first time since May. It was so good. You know we're getting a new Panda. It's open. Yeah, it's open in the. Yeah, which is open? Panda's open.
And we're doing a new one where Wendy's was.
Yeah. Mm-hmm. That's like a for real one, not a baby panda. Oh, where's that one going to be?
It's where Taboo used to be.
Where?
Remember that place that they had Taboo?
Oh yeah.
Coffee shop?
Oh.
Yeah.
Oh. So we're going to be learned individuals and fat individuals. That's what we're saying? Okay. When does myelination occur during development? When does myelination occur during human development? It's going to begin pretty late, 7, 8 months. And it's not going to be completed really until a few months after birth. So when you get that brand new bouncing baby, they're not really bouncing unless you drop them. And then they don't bounce that much. But they don't really work that well either. Not only do they not have a lot of strength and coordination with their neck muscles, they don't have a whole lot of coordination, period. Adrian, you've been learning a lot about handling? Yeah, I noticed you look a little sleepy over there. Yeah. But it's so cool, the built-in reflexes that babies have. If you can be around a brand new baby, I mean just a couple of days, couple of weeks old even, all you have to do is just rub your finger on the cheek of that baby, and that baby is going to turn its head and just automatically going to start sucking. So you've got some information being transmitted. You've got some myelination happening. But I think the myelination in the nerves for the neck muscles doesn't exist because that head is just, you got to hang on to the head.
You know online lately that's going around? Like the newborn, like brand new newborn, like the nurses have it and it's like standing. I mean, like she's kind of holding it, but it's like kind of.
No,
I don't know.
I have to see that. Brand new babies I've seen just don't work. Their digestive tract works, but that's about it. And their lungs, because they can cry. Whoo, they can cry.
She like had her hand like on its chest and kind of was like moving its legs and stuff like while
And
it
their
was
legs are
like,
pretty
wow.
strong. Like, I was really surprised by that. Like,
Well, and the other thing too, their grip.
yeah.
If you've ever been around a baby and they grab your hair, you can't get them to let go.
I think it's interesting because, like, comparatively, like, we don't have that grip strength anymore. Like, and there's a couple of reflexes that they have that I find kind of weird. Like, it doesn't make sense, like, evolutionarily anymore, I don't think. Like, the one where they kind of, like,
Oh yeah, they're just trying to grab something because they're falling. I like the reflex when they're in the dark and you turn on a light. It's almost like a startle reflex. I used to do that too much and I get in trouble. Anyway, all right. Can a person survive without myelination? And the answer to that is no. Multiple sclerosis. This is a debilitating disease. A lot of people think it's muscular, but it's really nervous or neuromuscular, in the sense that you have an autoimmune condition in which your body attacks the myelinating cells of your body, specifically your oligodendrocytes. So are you preventing the transmission of the action potential? No, you're not. But what are you doing to the action potential when you delete these oligodendrocytes? And you delete the myelination. You're slowing it down 100-fold. So you lose the coordination. You use the coordination. I mean, walking, for instance. People that work in robotics, they had such a difficult time programming robots to walk, bipedal robots to walk. Because what is walking to a human? I mean, babies don't just automatically do it. Well, most babies don't. But what is walking? Losing your balance, falling, and catching yourself before you fall. You try to train robots not to fall, and now you're saying, okay, go ahead and fall. And the robot's like, does not compute. But there's a lot of coordination that goes on. Imagine walking, but your coordination is slowed down 100-fold. That's why when you see someone with multiple sclerosis, it's usually small steps, very jerky steps, careful, because the coordination is lost. Because the signals simply can't get transmitted fast enough. Now, I've put this graphic up a number of years because it doesn't make sense to me. I'm a logic kind of driven person, a data driven person. So where you have red, this is the highest risk of multiple sclerosis. Purple, probable. This brown, low risk. Yellow, probable, low risk. Other risk, I guess lower type risk. Why in the world are we in such bad shape, except down here in the south? I'm thinking we're down here almost in the brown even. Ha, we're in good shape. My hypothesis, just because of what I like to eat, fried foods protect you from multiple sclerosis. I don't think it works. But it's not, it's not longitude. It's not climate. You even got some stuff down here off of Australia. So I don't know. So let that percolate in your mind for a little while. And I've not been able to find anything that's really sort of helped me come to grips with why in the world. I would think that if it was ancestral and from an evolutionary or genetic standpoint, it would seem to reason that our ancestors came from the Inuits that crossed over here in Alaska, so why don't they have the same risk? I don't know. Well, now you do have some of the Germanics over here. Certainly a lot of Europeans. I'd say there are a lot of Italians as well. And over here in Italy, we've got this green. So again, there's not been one thing that I've said, oh, because you can always say, yeah, but what about this? Yeah, but what about this? Several semesters I've looked at this and it just, I don't know. It's just a puzzle. It's always puzzled me. Okay. So we've propagated our signals along an unmyelinated axon and a myelinated axon. Sped it up really, really fast, 100 meters per second plus. Well, we don't just have directly wired neurons going from our central nervous system to every part of the body. In fact, we have interconnected neurons. But they're not physically touching each other. They're separated by space. So how do we transfer that electrical signal from one neuron to another or from a neuron to a muscle cell with no physical connection? How does that happen? This happens at a junction that's called a synapse. And what's going to happen, sort of in a nutshell, is we are going to transduce our signal. We're going to change the type of signal from an electrical signal in this neuron to a chemical signal that is going to be able to cross space. Guess what kind of transport mechanism we're going to use for this chemical to cross this space? It's just diffusion. It's empty space. This is going to be diffusion that's going to happen. And when the chemical gets to this second neuron or a muscle, the target cell, you're going to have receptors for those chemicals that are going to be able to assist in recreating the action potential. So do you see how we're changing or transducing electrical to chemical back to electrical? And that's what's going to happen at these junctional complexes we call a synapse. Now, in the nervous system, we can synapse neuron to neuron, either axons to dendrites, and that's going to be the most common type of synapse we see in our nervous system. But we can also synapse an axon to the cell body or an axon to an axon. Because remember, our signal's being sent by an axon, so that's why they all start axo. Axodendritic, somatic, axonic for those respective places where we're going to see those synapses. So here's our axodendritic, axosomatic, and that connection right there is the axoaxonic. You see, this axon is actually going to the cell body, but this one is sort of heading it off at the pass, if you will. And these are the means with which we can regulate the signals and have a higher degree of fidelity and control of the signals that are sent between cells. Now for our classical synapse, we are going to use a neuromuscular junction. It's also called a myoneural junction. Myo is a prefix meaning muscle. And this synapse is going to be between a motor neuron and a skeletal muscle cell. Now, skeletal muscle is going to be our next chapter, so we're already starting to sort of move in that direction. And so with our synapse, we're going to have this gap, this physical space that we call the synaptic cleft. And relative to the synaptic cleft, we have the signaling cell and we have the receiving cell. This is the presynaptic cell. The signaler is before the synapse, so that's the presynaptic. The receiver is the postsynaptic cell. You see how we're designating those? And again, I keep tapping this table, and this one is our pre and post with this space in between. It makes a nice little visual. Okay? So here we have the very end of our alpha motor neuron. And in these type of synapses, the end of the axon spreads out. It looks like a little button. And so it's often called the terminal bouton. And at each one of these termini, you can see that the axon interdigitates itself within the plasma membrane of the muscle cell. And that muscle cell actually folds itself around, so that the yellow you see in this illustration is our presynaptic membrane, and the folded membrane of the muscle, that is our postsynaptic membrane. Now, anytime you see folds, what are you going to be thinking about? Remember I said that diffusion was how the chemical traveled. So think about chemicals, think about diffusion, and think about this folded membrane. What are we trying to accomplish here? Why do we have these folds? Speeds the rate because of what have we done? Increase the surface area of the receiving cell. And that increased surface area is going to help us speed up the detection of our chemical signal to make diffusion more efficient and more rapid. Okay, so we're going to start with our presynaptic neuron. Action potential was sliding down the axon. It's myelinated, so we're traveling at over 100 meters per second. Now, our terminus, the action potential hasn't gotten here yet, so we're at minus 70 millivolts. We're at rest. And in the very end of our motor neuron, we have all of these little vesicles that contain the neurotransmitter, which is specific to stimulate our skeletal muscle cell. And we're going to go ahead and put some specifics here. That neurotransmitter for skeletal muscles is called aceylcholine, and our abbreviation is ACH. Those neurotransmitters, those are our chemical signalers, the chemical messengers that are going to cross the synaptic space and bind to the receptors on that skeletal muscle cell. Now, these vesicles are not only present in the cytoplasm at the end, many of these vesicles are docked. They're already fused with the plasma membrane, just ready to be released. I mean, it's like a runner in the starting blocks of a sprint, just waiting for the gun to sound. Because this is going to happen really, really quickly. Now, we increased the plasma membrane. So we've got increased surface area for the detection of the signal. What is another way that we can increase the rate of diffusion? We did this with our sodium and our potassium in our neuron already. But for our neurotransmitters, how can we increase the rate for diffusion to happen? Create a very, very steep gradient. We're going to release the neurotransmitter, so already there's going to be zero concentration on this end and some on this end. But if we want to have a really steep concentration gradient, we're going to have a bucket load over here and 0 over here. Check it out. There are about 300,000 plus vesicles in the end of our axon. Each vesicle is going to contain 10,000 molecules of acetylcholine. Can you do the math? That's a lot of zeros. So do you see how steep that gradient's going to be? I mean, talk about jumping out of an airplane. It's straight down. So we've got the increased surface area. We've got this huge gradient of neurotransmitters. Diffusion is going to happen like a tsunami that's going to wash over so that we can transduce that signal to the recipient cell. So we've got our acetylcholine vesicles docked. Our action potential is sliding down the axon. Sodium voltage gated channels open, depolarization. Potassium voltage gated channels open, repolarization. When we get to the end, we've got one more partner that we're going to enter into the mix. A voltage gated calcium channel. Remember, calcium was another one of those ions that's highest concentration outside of the cell. So as the action potential slides down, it's going to open our voltage-gated calcium channels. Calcium is going to follow its concentration gradient and flood into the end of the axon. Now think back to some of your cell biology that you had. Do you remember your second messenger signaling cascades? Again, like the domino effects. This is going to signal this is going to signal this, and on and on and on. Calcium coming into the end of that neuron is going to start a signaling cascade because calcium is going to bind to a protein called calmodulin. Calmodulin's like the car, calcium is like the key that starts the car. When calcium binds, calmodulin turns on. It is then going to start a cascade. And this cascade is going to involve calmodulin causing the phosphorylation of other regulatory proteins, regulatory signalers, and the end product of calcium influx and calmodulin activation is going to be the release of our neurotransmitters into the synaptic space. If calcium cannot flood into the neuron, you will not have your neurotransmitter released. So calcium's not just for bones. It's also important for the nervous system. So drink your milk. Where else do you get calcium? What are other sources of calcium? Don't say ice cream. Dairy. Cheese. That's kind of dairy, I guess. Is there a vegetable that has calcium in it? I want to say broccoli or asparagus or something like that. Maybe it's broccoli and that's why you put cheese on it. Give you even more calcium. I can't remember. So calcium, here's our voltage-gated calcium channels. Calcium influx. Calmodulin becomes activated. It's going to activate this signaler called a protein kinase that phosphorylates regulatory proteins. That's going to lead to the release of our neurotransmitters. So now we've got our chemical messengers released into the synaptic cleft, this space. And this space is going to be fairly narrow, only 20 nanometers. So what are the ways in which we have sped up diffusion? Increased the surface area of the receiver, created a ridiculously steep gradient. Now we're going to shorten the distance between the 2 points, only 20 nanometers. And this 20 nanometer space is going to be maintained because of membrane receptors in the pre and membrane receptors in the postsynaptic cell, like the little loops and hooks of Velcro that are going to maintain a very precise distance. And it's within this 20 nanometer space that our neurotransmitters are going to rapidly diffuse. And when they reach the postsynaptic membrane, they are going to bind to a set of receptors that are now called chemical receptors or chemically gated receptors in the case of acetylcholine. Because on our skeletal muscle cell, we have an acetylcholine receptor. And we're going to see this in a couple of slides, I think, but go ahead and jot this down in the notes somewhere. In your skeletal muscle cell, the acetylcholine receptor is called a nicotinic receptor, like nicotine. A nicotinic acetylcholine receptor. Nicotinic, I see. And this nicotinic receptor is going to have to bind 2 molecules of acetylcholine to be activated. If it just binds 1, nothing's going to happen. It has to bind 2. And when 2 molecules of acetylcholine bind to this nicotinic receptor, that nicotinic receptor is going to open. Because in addition to binding acetylcholine, a nicotinic receptor is also an ion channel. And so when 2 acetylcholine bind and the nicotinic receptor opens, it allows sodium to come into the cell. And it allows potassium to go out of the cell. What does that sound like? Sodium coming in, potassium going out. What's that sound like? It sounds like the pump, but it sounds certainly like what we did with our action potential. And so when we start moving these ions through the nicotinic receptor, it's not repropagating the action potential, because what receptor do we have to have, or what channel do we have to get an action potential? Sodium voltage gated channel. This is not a sodium voltage gated channel. It's sodium and potassium. But what's going to happen is when we open that nicotinic receptor, ions change place such that our membrane potential gets more positive than -70. Now, a couple of slides ago before our break, we said that was a depolarization. But you remember in our graph, I said that little bump? I don't want to call that a depolarization. I call it a stimulus. That is our stimulus. That more positive change up until we get to -55 is called an excitatory postsynaptic potential, an EPSP. The key to the EPSP is the E. It's excitatory. So we're changing the membrane potential in this local environment where we bound acetylcholine to our nicotinic receptor. When sodium comes in, where is it going to go? Inside the cell, and once inside the cell, it goes everywhere, just like we saw with the sodium voltage gated channel. It's going to change the membrane potential, not just around the receptor, but in that local setting of 1 to 2 millimeters. And that's going to be the key. Because as these ions, sodium coming in, potassium going out, here you can see we've got 2 acetylcholine bound. Over here, this one's closed. Why is this nicotinic receptor closed? What's happening right here? We've only got 1 acetylcholine bound. We don't have 2 bound yet. When we talk about other types of muscle, smooth muscle, cardiac muscle, we're going to have a different type of acetylcholine receptor. And I just want you to understand that muscarinic receptors, and we're going to do quite a bit with these in the next chapter, but for right now, just introducing them. Muscarinic receptors bind acetylcholine, they just bind 1, but they're not ion channels themselves. They're signalers. And many of them signal through complexes called G proteins. So muscarinic receptors bind 1 molecule of acetylcholine, signal through the G protein receptor complex, and that will activate or inhibit, depending upon the tissue and the receptors, a different ion channel. So it's a series. And in the case of our cardiac and smooth, we'll see in smooth muscle, the activation of a muscarinic receptor closes the channel. This is going to depolarize and lead to a contraction. Whereas in cardiac muscle, it's going to open some channels, leads to a hyperpolarization, and is going to inhibit or slow down the cardiac muscle. All right? So again, don't sweat those details because we're going to get into a lot more details of that coming up down the road. So I want to go back to our nicotinic bound to acetylcholines. Sodium and potassium moved following their concentration gradients, and we made the membrane more positive. An excitatory postsynaptic potential. Now, wherever we have those receptors binding to 2-acetylcholine, that's going to happen. But immediately surrounding that terminal bouton, that neuromuscular junction, we're going to have other receptors also present that are going to be affected because of the cable properties. But in the case of our anesthetics we talked about earlier, blocking sodium permeability, well, in the case of our nicotinic receptors, certain less civilized tribes that have to be hunter-gatherers, they discovered that the chemical curare, which I believe is obtained from some plants, they put these on the end of their blow darts and they can actually paralyze animals. Certain animals that are arboreous, that live in trees, if they get hit with a curare dart, that blocks the nicotinic receptors, so all of their skeletal muscles are paralyzed. They fall out of the tree and they're fairly easy to catch that way. So curare directly binds to the nicotinic receptors in such a way that it prevents their activation because acetylcholine can't also then bind. So here's an illustration of our terminus. Here's our folded membrane of our muscle cell. The green are our nicotinic receptors. And I really wish I could delete that. I might have to Photoshop that little blue sucker out right there. I don't like it being there. Because on the periphery that you see right here, adjacent to our terminal bouton and our nicotinic receptors, we have sodium voltage gated channels. So whenever our nicotinic receptors allow for that excitatory postsynaptic potential, that stimulus that makes the membrane more positive, that membrane is going to be more positive than minus 55. And because of the cable properties, those ions are going to flow adjacent to it less than a millimeter away. It's going to change the local membrane around our sodium voltage gated channels to minus 55 or more positive. And so what's going to happen to our sodium voltage gated channels? They're going to depolarize. They're going to open. Since these are sodium voltage gated channels, what have we just repropagated in the second cell? An action potential. So we had an action potential here, transduced it into a chemical signal, detected it with our nicotinic receptor, leading to a change in voltage that was excitatory, that then excited our peripheral sodium voltage gated channels. Boom, another action potential in the second cell. This is going to depolarize to what level? Now that's when we open, negative 55. Remember our depolarization went up? What was that top number? Do you remember? 30. All of these sodium voltage gated channels are going to depolarize to positive 30. This sodium voltage gated channel is going to depolarize to positive 30. When you get an action potential, it's the same strength no matter what, no matter where. And so now you have just repropagated your electrical signal that's going to spread all along the surface of that skeletal muscle cell. And that's how we propagate an action potential from a nerve to a a muscle, or from a nerve to a nerve, or a nerve to a gland.